We should get going. Good to see everybody. I hope you enjoyed your weekend. You have midterm number 4 coming up 1 week from today. And as usual, it's going to be in during the laboratory portion of the class, and it will have the following topics on it from lecture. It's going to have signal transduction, it's going to have chromosomes, mitosis and meiosis, and the chapter we're on now, which is genetics. I'm going to put a little asterisk there because we're probably not going to finish the entire genetics lecture by Monday, and so the exam will only contain questions on the parts of the genetics lecture that we have covered. As far as lab is concerned, there was a couple of labs we did on photosynthesis. So a question or two from both of those labs. There was a lab we did on bean respiration. You can expect a question from that lab. You did a lab on chromosomes. Which most of what that lab was reviewing was concepts related to mitosis and meiosis. So in a way, it's almost the same topic as what we covered in lecture on those. And this week, you're going to do some labs on genetics, which are actually just some worksheets where you're going to work some genetics problems during the laboratory portion. And so anyway, those laboratories this week actually are essentially overlapping with the genetics lecture that you'll be getting in here. Clear enough. And a couple heads ups. Remember that every Monday you get a weekly quiz on the last 2 laboratories that you did. And so when you come in today in laboratory right at 2 o'clock sharp, you'll get a weekly quiz on what we do last week, bean respiration and the chromosomes lab. So be ready to answer questions on those at 2 o'clock today. As part of the genetics labs this week, I'm going to be covering part of the genetics lecture. So bring your lecture handout from genetics with you to laboratory this week. Alrighty. All right, well, let's dive right back into genetics. I should do a brief recap of the major concepts. When Gregor Mendel was figuring out the laws of genetics, he made a differentiation between genes and alleles. A gene is something that controls a trait. For instance, as an example, there's a gene that controls flower color in the pea plants he was studying, but there's more than one version of that gene. There's what's called the dominant allele, and there's what's called the recessive allele. And for the flower color gene, it happens that the dominant allele was the one that gives purple flowers, and the recessive allele was the one that tries to give the plant white flowers. And the reason why they call them dominant and recessive is that the dominant allele, the purple one in this case, controls the trait whenever it's present. And whereas the recessive trait only controls the trait if there's no dominant allele around. You only get white flowers if there's no purple allele within the plant. And lastly, review-wise, Mendel said we should just use one letter to represent both alleles. And what he normally said is let's use the capital letter to represent the dominant allele and the lowercase of that same letter to represent the recessive allele. And usually he picked the letter that described the dominant allele's trait, like since it's a purple flower, capital P means the dominant allele, and lowercase p would mean the recessive allele. And since we all have two parents, we all have two copies of every gene. You inherit one copy of every gene from your mother and one copy of every gene from your father. And so for every trait, you have two alleles. And which two alleles you have, either two of the dominant or two of the recessive, or one of the dominant and one of the recessive, is called your genotype. Here's a list of the possible genotypes. Homozygous dominant is big P, heterozygous, big P, little p, and homozygous recessive, little p. And the phenotype are the physical traits that you have, which of course are based on what alleles you have, based on your genotype. And remembering this definition of dominant is a dominant allele controls the trait whenever it's present. So if you're a big P plant, you have purple flowers. And if you're a big P plant, you also have purple flowers because big P for purple is the dominant allele. It dominates over the white colored allele. Whereas if you're little p, homozygous recessive, then and only then do you have your phenotype white flowers. All right, and then we started talking about things called Punnett squares, which is a systematic approach for doing crossings or matings of organisms to see what sort of genotypes and phenotypes the offspring will end up with. Here's one of Mendel's crosses. This purple plant is big P, and this little one's little p. Notice all the offspring are purple colored. Let's see. There we go. And here's how you would analyze this crossing using a Punnett square. To write a Punnett square, you make well, you write both the parents, first of all, and their genotypes. And you draw a square like this. And under each parent in the columns here and in the rows here, you write what sort of gametes, what sort of sperm or eggs that parent could produce. And remember that when you make your gametes, they're haploid, so they'll only get one of your two genes. But for that plant, since it's homozygous dominant, it doesn't matter whether it gets the maternal allele or the paternal allele that plant inherited from its parents. It doesn't matter, it's going to be big P. And likewise, when that white plant, it inherited little p from its mom and it inherited little p from its dad. And so when those get split apart in meiosis to make its egg cells, it doesn't matter whether the egg cell gets the maternal little p or the paternal little p. The egg cell is going to be carrying little p. So anyway, you construct the Punnett square based on the gametes that the organisms can make. And then you fill in each square within the Punnett square with the allele on top and the allele to the left. And this tells you the genotypes of the offspring. And so notice that all of the squares within the Punnett square are big P, little p. And so that tells you 100% of the offspring are going to be big P, little p, which are purple plants because they have the big P dominant allele. And so this Punnett square would predict that all of that generation, which is called the F1 generation, would be purple. And that's exactly what Mendel saw in his F1 generation. All these guys are purple phenotype, and their genotype is big P, little p. And then Mendel went on to say, okay, what happens if I cross this F1 generation with themselves? Remember, they're all big P, little p. If I mate them with themselves, what do I get? And here's kind of a preview of what the results that he got are. But let's look at that with a Punnett square. Oh, so there's the genotypes of the first cross. Parental generation, offspring generation is there, big P, little p. So now we're mating the F1 to get the F2. Do it the same way. You draw the parents with their genotypes, make your Punnett square, fill in the gamete types that each parent, whoa, go back. There we go. Fill in the gametes that each parent can make. Half the sperm this one makes will carry the big P allele, half the sperm will carry the little p allele, and the same with this parent right here with her eggs. Half the eggs will carry big P, half the eggs will carry little p, like that. And so if you fill in the table, it shows this as the offspring. And you fill in the phenotypes from those 4 genotypes. That's what you get. It would predict that 3 out of 4 would be purple and 1 out of 4 would be white, which is exactly what Mendel saw. Sound all right? All right, and as I mentioned, this week in laboratory, you're going to be doing some worksheets, and so you'll get a chance to work lots and lots and lots of these Punnett squares. Okay, now maybe I should take a moment here just to talk about what the results of a Punnett square mean, and exactly what they don't mean. They aren't fate. They don't tell you exactly what the offspring are going to be. In other words, this Punnett square would not say the first offspring is going to be purple with a genotype of big P, and does not say the second offspring is always going to be purple with a genotype big P, little p, and the third would be not big P, and the fourth is going to be little p, little p. It doesn't tell you exactly what the future is going to be. And it doesn't even tell you exact numbers. It doesn't say that 3 out of 4 of the offspring will always be purple, and 1 out of 4 of the offspring will always be white. Well, so what is it telling us then? It just tells us that the chance, it tells you the probabilities of any one offspring having a particular genotype. It says that, well, maybe I can concentrate on phenotype here for a moment. Since 3 out of the 4 boxes would be purple flowers and 1 out of the 4 boxes would be white flowers, it says each and every offspring, each and every seed that's produced from this mating has a 3 out of 4 chance of being purple and a 1 out of 4 chance of being white. A good analogy is a coin toss. You probably know that if someone says, what's the chance that I'm going to throw heads if I toss a coin? You could say, well, it's half, 50-50, or 50% that you'll get heads on a coin flip. But that doesn't mean if you flip heads, the next flip has to be tails. That's not what that means. And it doesn't mean that you can't flip heads 5 or 6 times in a row. It's kind of unlikely, but you could do it. This just means that on each and every individual flip, you have a 1 out of 2 chance, a 50% chance of getting heads. So this says for each and every offspring, no matter how many offspring you have, each and every individual offspring has a 3 out of 4 chance of coming out purple and a 1 out of 4 chance of coming out white. And here's Mendel's actual data from his F1s mating to make the F2 flowers. And if you notice it, that it is about 3 out of 4 purple and 1 out of 4 white. But maybe just as important, notice that they're not exactly what we would, it's not exactly 3 out of 4 purple and 1 out of 4 white. Because just like the coin toss, there's going to be a little bit of random variability to it. But the principle that governs coin tosses and things like this is the larger numbers, the closer they are going to be to the expected ratio. If you flip a coin 3 times, it's not unlikely that you'll get all heads. Or I should say it's not super likely, but you can do it. But if you flip a coin 1,000 times, it's extremely unlikely you'll get 1,000 heads in a row. So the larger your sample size, the closer you come to the expected values. All right, and a little note down here that this little fact is going to come into play a little bit later on. If you have the chance of 2 separate things, and you want to know what's the chance of both those events happening, you multiply the probabilities of each one. So if I say, what's the chance of me flipping a head on a coin and then throwing a dice and have it come up 1. Who can tell me what the chance of that is? If I say, what's the odds if you flip a coin, get heads, and then throw a dice and get 1? Well, what's the chance of getting the heads on the coin flip? 1/2. What's the chance of throwing a 1 on a dice? 1 in 6. So if I say, what's the chance of both of those occurring? It's 1/2 times 1/6 over 6, which is what? 1 out of 12. There we go. So yeah, so it's just saying whenever you have 2 probabilities, multiply them to get the probability of both events occurring. Awesome. Okay. Let's see now. Oh yeah, so there's that cross right there. On the next part of your handout, I listed some common types of Punnett square crosses. As we go through this chapter on genetics, we're going to see a lot of these particular crosses show up over and over again. So it probably helps if you memorize some of these common crosses. The one that we just seen up here is called the monohybrid cross. Hybrid means heterozygous. And so if you have a heterozygous individual crossed with a heterozygous individual,